All right. Let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. Guten Tag. 안녕하세요. Hello all. Welcome and thank you, SynBYSS people, for joining us, the 75th seminar, and I call it a JBEI Day. I am in Germany. My first international trip this year started this week, and I have already given 2 talks in Europe this week. There has been strikes in Germany transport workers, making my trips a little bit complicated. However, there's nothing to worry about in life, because if the problem is solvable, no worry is necessary. Just try hard to solve it. If not solvable, more importantly, not controllable, I can just do nothing. Just think about plan B, C, etc. So let's be optimistic and hope for the best. Okay. It is my tremendous honor to introduce very briefly, although I'm not sure I can introduce him briefly, our pioneer speaker, Dr. Nathan Hillson. He is a computational senior staff scientist as well as a director of synthetic biology informatics at Joint BioEnergy Institute, JBEI, and Lawrence Berkeley National Laboratory. Among many roles he has, my favorite one is that he is an EBRC member of the Biosecurity and Biosafety Working Group, of which I am also a member. He provides very often very important views and insight during our monthly meetings, and actually I learn a lot from him. And Nathan, I truly thank you for your time and for your contribution to the scientific society and community, especially by agreeing to speak for young people in response to my short notice and invitation due to the unexpected cancellation of originally scheduled speaker. And life is full of unexpected events, including the strike I'm currently facing, and there are always, however, alternatives and sometimes much better solutions if we seek the heart. And Nathan, thank you again. The virtual podium is all yours now. Thank you.
All right, thank you, Tae-sok, and thank you for the invitation. I'm glad that I was able to participate, and thanks in advance to Logan. I'm looking forward to see your presentation. I think maybe what I'll just do in a couple of minutes is maybe talk more specifically to early career scientists that are thinking about their future career trajectories. And I won't have a lot of time, but maybe just a couple of bullet point, types of level types of topics, and I'd be happy to follow up with anyone, outside of the context of this seminar. So maybe just a couple of things that I think I'll note for early career folks. And one of the first things I would suggest to everybody is to actively engage with any type of mentoring programs that your universities or institutions or other types of communities offer. By mentorship, I generally mean not your direct boss or supervisor. That's more of a coaching relationship. What I mean by mentoring is somebody who's kind of arm's length away from you, who doesn't have a conflict of interest in giving you advice. So you want to have somebody that you can confide in, that you can trust, somebody that's actually, able or willing to say that the best thing for your future career development is to actually quit grad school or to leave your current job and go somewhere else. I think it's really important to have that type of a mentoring, type of relationship. And if and when you also have the capacity to do it in reverse, and you have the opportunity to mentor someone else, that's a really great thing. So I'll make that type of a statement. The other kind of suggestion I would have is always be on the lookout of certain types of opportunities around fellowships. These can be kind of like, short term, on the time scale of a week or two. It could be an internship. I myself have greatly benefited from at least two different fellowship types of programs. Quite a few numbers years ago, Megan Palmer and Drew Endy organized the Synthetic Biology LEAP program, Leadership Excellence Accelerator program. And the cohort that I was in is probably going to have a lot of names and faces that would be recognizable to people. And I think that was just a fantastic experience, not just for meeting people, but kind of understanding and learning new concepts. And another program that I think was really great was there's now John Hopkins is supporting it, but there's an emerging leadership or emerging leaders in biosecurity initiative, LB. And that's basically that fellowship is why I'm able to participate with TASOC, for example, in these EBRC biosecurity and biosafety working groups. It's nothing that I would have normally encountered in my day-to-day job. But I think through that program, I had an awareness and a different way of thinking about certain types of things. So that would be another recommendation. A third type of a thing is more going back to coaching and talking with your own supervisor, is that it's best, in my opinion, to be as transparent and direct as possible. One anecdotal story is that in my postdoc lab, people knew, and I'll put "knew" in quotes, that if you told our advisor that you were leaving academia, she would effectively disown you, because why should she invest any more of her time or effort in you if you're going to be leaving her subfield? So there was this big fear that if she knew that you wanted to go to industry, then she would no longer kind of invest her time and effort into you. To the extent where at Stanford, Genentech would send buses maybe like once a month to take people from Stanford to go to Genentech to basically learn about the company and take tours and things like that. And I knew that I wanted to go into industry, and I thought that maybe it'd be interesting to hear what Genentech was doing. And I showed up on the bus, and to my surprise, 4 other of my lab members were also on the bus, but no one told each other that they were going, because no one wanted anyone else to know that we were considering a career path that wasn't industry. But not too long after that, I mean, I had kind of an open and frank discussion with my advisor that I was interested in wrapping up my postdoc, and she herself had a startup company, and I was thinking, I saw this position that was open at your startup company. I think I'd be a really great fit. What do you think? And I think that was an important conversation, because after having that conversation, my advisor recognized that I was thinking about moving on, and that I was interested in industry. And at the time, she said, I don't think you're quite ready yet. There's a couple of little things to wrap up, but I'm sure if you really want a job at my company, then we can make that happen. But I think not too long after that, she was also pointing out other job opportunities to me, which actually ended up leading me back to the national lab. So I'm now a scientist at Berkeley National Lab. For those of you that aren't as familiar with the United States Department of Energy's national labs, where I am is at Berkeley National Lab in Berkeley, California. It's just up the hill from UC Berkeley's campus. The national lab system is somewhere in between an industry environment and an academic environment. We still do completely, open science. We publish basically everything that we do. We still apply for grants. We still have to publish and do all of those types of things. We tend to work on very big team science projects with infrastructure that aren't generally accessible to individual investigators at universities. So, and we were just talking before this meeting started that I think, if you look at individual investigator, type of a lab work, we're never going to be as effective or efficient as TASOC or others, for example, just because we're going to be much more expensive, and that's not kind of really our sweet spot. But once you get to really large projects, that's where we kind of excel. And it's also, great to engage with academics or with industry. So we have lots of collaborations with other types of groups. And I think, the national labs can be a really great career trajectory for people. So in addition to just considering, classic, academic positions or industry positions, you might want to consider places like national labs or other big research institutes as something that's just slightly different than academic types of places. But I think I'll leave it there and make sure that Logan has plenty of time to give his presentation.
Absolutely wonderful talk. I 100% agree with you. I believe big problem we currently facing, including climate problem, food security, whatever, all the problem only solvable by collaboration. And then that should be big collaboration. I, have absolutely, wanted to work with many people in the society, including your lab, your, entire, natural lab people. And second thing I really appreciate, you just mentioned your experience, and I also got very, very good advice from all my own advisor, Kristara Preder and also Chris Foy. And many advisors genuinely care about young people's growth and, expansion of their career. And even I exchanged email with Kristara today, even though I'm already established PI, but whenever I have problem, I just send email to her and she quickly respond to me. So I really, really appreciate my advisors. And then you also touch very important point about mentoring. And there are multiple programs, in scientific society, including American, Chemical Society, ACS, AICHE, SIMB. Actually, I'm also serving as mentor since 2021, just to help the young people. So that is, you need to utilize those things because we want to help, but if you do not reach out to us, we cannot just help you. So absolutely every point you just mentioned is fantastic point. I hope young people utilizing, older generation. In that way, you become the next future leaders. And thank you so much. That's absolutely wonderful talk. Thank you.
All right. Thank you, Tae-seok.
Yeah. All right. So now the main speaker of today with a longer introduction. Logan Collins is a synthetic biologist, author, and futurist. He is also a PhD candidate in biomedical engineering at WashU. He began engaging in scientific research when he was a sophomore in high school, very early in his career. Logan's research projects over the years have included designing an aggregating antimicrobial peptide and bacterial conjugation delivery system as a new way to tackle antibiotic resistance, developing a nanotechnology-based contrast agent for X-ray imaging of brain tissue, and leading supercomputer simulations of SARS-CoV-2 burden to better understand COVID-19. He is now working on a novel delivery vehicle for polygenic CRISPR therapies, which will have broad applicability for treating complex ailments such as aging, cancer, immunological dysfunctions, and more. Logan has spoken at TEDx Mile High, the Da Vinci Institute, and the Princeton Envision Conference. He's also a published science fiction and sci-fi poetry author that I saw very interesting. Logan passionately advocates for applying interdisciplinary solutions to global challenges and leverages both the arts and science to help build a bright future. Logan, thanks for your time today, and I'm excited to listen to your talk, and please take it away. Thank you.
Thank you very much, Dr. Moon. I'm very pleased to speak here today, and I really appreciate your wonderful introduction. With that, I will share my slides. So today I'm going to be talking about my PhD research on developing a hybrid viral vector consisting of multiple viruses that are covalently linked together as a platform technology for delivery of multiple genes to the same cell. This technology will have broad applications across many different human ailments, and I'm really excited about how it can be applied to CRISPR, or how it can be used in the context of CRISPR to solve a variety of biomedical challenges. So I'd like to mention just briefly that this is still very much a work in progress. So I am right now in the midst of developing this technology. Nonetheless, I've made some very interesting and exciting strides that are certainly indicating that the technology has a bright future. So now I will begin by just talking a little bit about the background and significance of my work. So as mentioned, I'm using multiple viruses. So I'm specifically using adenovirus, which is pictured here, alongside AAV, or adeno-associated virus. I should mention that the reason that adeno-associated virus is named as such is because in the wild, adeno-associated virus requires adenovirus to replicate. However, this is sort of the extent of their natural association in the context of infectious disease. What I'm doing is for gene therapy. So it should be noted that the viruses have their replicative abilities ablated, so they no longer are able to replicate and cause human disease. Instead, functioning as gene therapy vehicles, essentially gene therapy vehicles which can help treat human disease. Adenovirus and AAV are both viral vectors for gene therapy. Viral vectors are a leading technology for gene delivery in a therapeutic context, and adenovirus and AAV both are some of the leading gene therapy vehicles. In addition, both have an immense potential for delivery of CRISPR-Cas gene editing therapies, which, as I'm sure many of you have heard, over the past decade or so, has been experiencing something of a renaissance in the context of gene delivery, gene editing, and also other applications such as plant biotechnology and so on and so forth. All right, so I'll go briefly over this slide because I know we have somewhat limited time. So I just want to mention that each of these viruses, adenovirus and AAV, has certain benefits and drawbacks. There is a trade-off in terms of capsid or immunogenicity uh, and transduction efficiency. So AAV transduces cells less efficiently than adenovirus, and so you have to use much higher doses of AAV when treating diseases. However, adenovirus has a higher immunogenicity and can be used at lower doses. So in this way, although adenovirus, oftentimes people may comment that adenovirus is more immunogenic than AAV, and so it could be a problematic vector, it actually has been used in a variety of clinical contexts, and one of the main advantages there is that the lower doses needed for transduction sort of balance out the immunogenicity. So both viruses have ways that you can engineer their capsids to target them to particular cells, to further decrease immunogenicity, and to confer other beneficial properties. The AAV has a very low DNA packaging capacity of about 4.7 kilobases, while adenovirus typically has a high packaging capacity of around 8.5 kilobases, but through some somewhat challenging engineering can be modified through additional deletion of genes to package up to 36 kilobases. There's also a difference in the episomal gene expression, so that's where the viruses sort of persist in cells just as circular DNA pieces that float around. And AAV, interestingly, can express genes for a year or more this way, whereas adenovirus, the episomal gene expression is only stable for a few weeks. So as you can see, there are many benefits and drawbacks associated with each of these viruses, but what if we could combine these vehicles through linking AAVs onto the surface of the adenovirus to make AdAAVs? This could enhance the benefits and mitigate many of the drawbacks while enabling multigenic or polygenic even gene delivery to individual cells. This is what I'm working on. And actually, before I move on to this slide, I'll just point to this diagram that I made to illustrate what an AdAAV conceptually may look like. And I'll show you some additional images later, but as you can see, the smaller capsids are the AAVs, and the larger core pink capsid is the adenovirus. The adenovirus has fiber proteins which extend outwards and should hypothetically protrude beyond the AAV capsids. And so just to give you a conceptual picture, I wanted to highlight that. AdAAVs have a variety of potential advantages. So, as mentioned, they could be used for multigenic DNA delivery, delivering multiple transgenes to the same cell. As illustrated here, we have fluorescent proteins that are red, green, and blue. In this conceptual diagram, you can see that each cell is expressing all three proteins. That's sort of the main selling point in my view. However, there are a variety of other benefits, potential benefits. So adenovirus fiber proteins may work synergistically with AAV capsids to facilitate strong transduction of targeted cells, even stronger potentially than adenovirus on its own. The reasoning behind this is that the adenovirus fiber knob may first bind to a cellular receptor, and then the avidity, the multivalent interactions from the many AAV capsids, in addition to additional adenovirus moieties, may facilitate sort of a enhanced stickiness of the AdAAV onto the cell surface, allowing for easier endosomal uptake. And then in the context of CRISPR, AAVs carry single-stranded DNA, and this could potentially be used as a donor template for CRISPR. So it's known that single-stranded DNA acts as a superior donor template for homology-directed repair, which is very common in the context of double-strand break CRISPR. And so one could envision that you could have an adenovirus encoding the Cas9 and several guide RNAs, and each different AAV encoding a different single-stranded donor template, thereby enabling programmable insertion of these transgenes into various locations in the genome with potentially higher efficiency than a double-stranded donor template. I'll also mention that there are a direction I'm currently pursuing in collaboration, early stages in collaboration with a startup company, is that at AAV with there's certain types of AAVs that have been engineered to cross the blood-brain barrier with very high efficiency, and I envision that the AdAAVs could be carried across the blood-brain barrier through a transcytosis mechanism um, by these AAVs that would sort of act as a vehicle to bring the entire complex into the brain, facilitating injectable, intravenously injectable AdAAVs that would have broad biodistribution across brain regions, potentially for treatment of neurodegenerative diseases, mental illnesses, etc. And then finally, AdAAVs should be easily amenable to site-specific and potentially cleavable shielding moieties. So earlier we talked about immunogenicity, or I talked about immunogenicity, and the immunogenicity may or may not be an issue depending on what dose is needed. But if it is, one could envision that you could attach a cleavable linker, in this case SpyCatcher, which I'll explain later, a spy catcher attached to a cleavable linker attached to polyethylene glycol or a similar shielding moiety. And then upon reaching a target tissue, some enzymes or something similar, low pH, might be able to cleave the cleavable linker, making it so that the shielding moiety, such as polyethylene glycol, does not interfere with the transduction of the ADAVs, while at the same time, during transport through the body, the shield still persists. So these are a lot of hypotheticals, but I wanted to point them out because I really do feel that there's a lot of potential for this technology to go in a lot of different directions that could have a lot of these positive potential advantages. All right, so I'll move on to what I've actually been doing here, and I'm going to talk about how I've constructed the ADAVs. So the current stage of my project is that I have successfully built and validated the ADAVs as nanotechnology constructs, but I'm at the very beginning stage of doing in vitro testing of cellular transduction, and I'll talk about that later. But I'm just beginning to transition from the engineering point to the testing point, so I'll just highlight that. But for now, I want to talk about SpyTag and SpyCatcher. So I mentioned SpyCatcher earlier. SpyCatcher and SpyTag are sort of an enabling technology for this project because what they allow is for site-specific covalent conjugation of protein molecules to each other. And in this case, I'm covalently linking AAVs onto adenovirus. Essentially, what SpyTag and SpyCatcher is that their SpyTag is a peptide, and SpyCatcher is a slightly larger peptide that react with each other in a very specific and highly efficient fashion to form a covalent isopeptide bond, as shown here. And this essentially allows it to act as a molecular glue, which is very useful for this project, as well as for a lot of other research out there. One of the things that needed to be done to prior to starting to stick these viruses together was to genetically engineer them to have capsid modifications, which introduce SpyTag. So what I and my collaborators have done is to, for one thing, engineer adenovirus to insert a SpyTag into the hexon, which is the hexagonal protein that makes up most of its capsid. And you can see on in this schematic that the SpyTag is in the hexon, and there's 720 SpyTags per adenovirus. Specifically, I'm using simian adenovirus 36 and AAV9. The simian adenovirus encodes GFP, or eGFP, as a fluorescent marker currently. And we have successfully rescued, upscaled, purified, and validated by protein gel this virus. In addition, collaborators at Baylor University, William Ligor's group specifically, have produced AAV9 with the SpyTag in the VP2 protein, which makes up approximately 1/12 of the total 60 capsid proteins in the AAV capsid. So AAV has a random, on average, each AAV has 5 VP2 proteins, but it does vary, so it's sort of a Gaussian distribution. And so the VP2 SpyTag only ends up on average on 5 of the AAV proteins in the capsid, which is nice because it reduces cross-linking, allows for higher titer production of the AAV, and minimizes any interference with the AAV's function. The AAV that I currently am using encodes TD tomato, a red fluorescent protein, and this is very useful since it contrasts with the GFP from the adenovirus. So one concern that has come up in the past for me is that, okay, so we have these viruses. If we cross-link them, they might just aggregate into a big mass. And I've actually found that this is less of an issue than one might think. However, I still did need to develop a novel protocol to facilitate the construction of AdAAVs without risking too much of that type of aggregation. So what I do is I take advantage of mass action kinetics to first treat AAV, the AAV SPY tag with a massive 300-fold excess of a bivalent form of spy catcher, by spy catcher. Then I remove excess by spy catcher with a size exclusion filter, and I recover the AAVs. The reason that you see here the AAVs minimally cross-link with each other is because it's just much more likely for a reaction between spy catcher or by spy catcher and spy tag to occur than it is for a reaction between by spy catcher, one spy tag on one AAV, and another spy tag on another AAV to occur. So essentially this saturates all the sites before they can really cross-link. So I remove the excess by spy catcher. Then I treat adenovirus with biotinylated spy catcher, just a small amount, just enough to cover a few, 50 out of the 720 spy tag sites so that it doesn't use up too many of them. This is for later purification, as you'll see in step 6 down below. And then once I have these two viruses prepared, I mix them overnight, incubate them overnight at 4 degrees Celsius. I use a 10-fold excess of AAV spy tag with by spy catcher. This has been I've found that this has been effective, and it also in terms of getting enough AAVs onto each adenovirus. However, what I've noticed is that there does tend to still be a lot of AAVs that are free floating even after the overnight reaction. And so that's something that's the main contaminant that I need to purify out. Before purification, I just saturate any remaining bi-spy catcher sites with a spy tag tethered to maltose binding protein. This is a commercially available reagent. It's fairly convenient. The reason for this is that it just essentially prevents any future aggregation from collisions between AAVs. And then finally, I use a affinity column called a streptactin column, which very weakly binds to biotin in order to purify the AdAAV particles, removing the free spy tag MVP and also removing free-floating AAVs. This has, as you'll see from my results shortly, this has proven to be pretty successful. One caveat I'll mention is that my titers, the recovery percentage of AdAAVs relative to input adenovirus is fairly low. And so I'm currently working on ways to optimize this further, improving the recovery percentage. I'm also looking into some alternative ways of purifying, such as sucrose gradients. So I'm going to skip over this because this is kind of a minor detail. Essentially, it's just how I construct the spy catcher biotin and put it onto the adenovirus. The streptactin purification, I mentioned that I'm using a I'm trying to optimize it to recover a higher amount of AdAAVs. You can sort of see some of that in this description of how many times I wash, what concentration of biotin I use, how many elution fractions I collect, so on and so forth. And I think next time I do it, probably next week actually, I'm going to be able to get a significantly larger number of AdAAVs out than I did previously. All right, so now I'll move on to the actual results. So I'll be showing you some imaging data. And what I used was, in collaboration with the core facility for electron microscopy, transmission electron microscopy, I used that as sort of my main quality control check. Because it really provides a very beautiful picture of how this, actually, let me just of how this works and gives us very clear idea of what's actually going on at the nanoscale. So just to mention, in terms of this being construction results, I want to include this slide again. I'm not going to repeat everything, but I'll just say that the construction results sort of include the outcomes from genetic capsid modifications, because that was a significant hurdle to overcome. prior to moving on to the rest of the project. And then now I'm going to describe 3 experiments that I performed in the process of developing my novel protocol for assembly and purification of ADAVs that sort of show the progression of how I was able to engineer these. So step 1 was just assembly test. This is basically everything but the purification. And in this case, everything but the purification. So what you can see here is that uh, there's a diluted form, 1 to 20 dilution of no purification after assembling ADAVs. Images were taken with negative stain transmission electron microscopy. And in this top row here, these are the ADAVs, and they look very nice. You can see that they do recapitulate what I envisioned in my diagram, and that they have many AAVs per adenovirus and so on and so forth. There is some variability I've noticed in the number of AAVs per adenovirus on a batch-to-batch basis as well. These were some exceptionally good ones from this particular batch, but I have been able to get similar results in other batches also. The lower row is my control. So you can see sometimes there's maybe some kind of electrostatic association between the AAV and the adenovirus, but it's not anywhere near as dramatic as the ADAVs. The control is just using a spy tagless or no spy tag adenovirus. So the AAV still has spy tag, but no spy tag on the adenovirus, so they don't conjugate. And this was sort of my first validation test. My second test was to ensure that the purification method would operate appropriately. So I started to develop this purification method from scratch. Essentially, I have I have the top row here, which is the experimental, and then they have the pre-purification assembly mix, where there are a bunch of AAVs floating around, a bunch of contaminants. And then in post-purification, you have maybe less AAVs, but most of the impurities are removed. And I have 2 different examples here. And then in the control, it's the same thing essentially, except that I use ADAV with SpyCatcher instead of SpyCatcher biotin. So no biotin, no affinity to the streptactin. And so the ADAVs just come out in the washes, and at the end, you don't end up with any ADAVs. And across many fields of view, it's like this. So you don't see any ADAVs because they got washed out because they weren't sticking to the column. And that's the control, thereby illustrating that the purification method works. So then the third experiment is essentially the same thing, but a larger batch with just the experimental. So I have my pre-purification assembly mix here, lots of AAVs, lots of adenovirus, lots of ADAVs, and then 2 examples of fields of view from the post-purification. You can see that there are more ADAVs in the post-purification here since I started with more. And then I also have some nice zoomed in images of some of them. So it's very nice because essentially what this does is it just removes all those free-floating AAVs and other contaminants, which allows me to then pursue further experiments. So I alluded to this earlier. This is the qPCR-based titer of the viruses. So I was able to get a small amount of ADAVs out of this purification method. I mentioned a variety of ways for improving the yield, including adjusting the parameters of the experiment sum, which I actually think will make a significant difference based on what I've seen so far, as well as potentially pursuing a sucrose gradient method as an alternative, or even other affinity methods that might be more efficient than the biotin-streptactin interaction. So this is and the reason I use qPCR for this is because it allows for titering of both the adenovirus and the AAV in the same prep, because they're obviously conjugated together. You can't just measure the concentration of DNA after breaking everything open, as I often have done with the adenovirus. So this allows for both viruses, the number of both viruses in the in the prep to be measured, even though they're covalently associated, which is very nice. All right, so now I'm going to talk a little bit about some next steps. I will say that I've also done some interesting work with some sort of offshoots of this project I won't talk about today, but I will mention that there are some other directions, such as the blood-brain barrier crossing at AAVs. There's also something I'm working on involving phage that's very interesting. But for now, I'm going to focus on sort of the core AAV project. And what I want to talk about first is sort of the very beginning of my cellular transduction assays. And I put this in next steps because it's currently a pretty early stage experiment. This was the data from the very first time I did this experiment. And I think that there are definitely some ways that it could be improved next time I do the experiment. But essentially what happened was that the ADAVs definitely do transduce the cells, but it seems that the adenovirus gene expression is a lot more frequent than the AAV gene expression, which is AAVs are in red, TD tomato. And there are a variety of reasons for this issue. The one listed here, in addition to the one listed here, is that different cell types can essentially lead to different types of intracellular trafficking of AAV. So even if the adenovirus brings AAVs into the cell, it's not guaranteed that the AAVs are going to express a gene because of the way they traffic intracellularly. And so it may be that choosing different AAV serotypes could be better for different therapeutic contexts in the future. So different AAV serotypes, the one I'm using, AAV9, tends to be better in neurobiological contexts. So I'm thinking about maybe using a cell line that is neuron derived next time I do this experiment. And AAV2 by contrast, which I don't currently have an AAV2 spy tag, but I could make one. AAV2 is better for like the liver, for example, hepatocytes and so on and so forth. So what's listed here is an interesting hypothesis that I had that AAV might be limited in its gene expression because it could remain trapped on the adenovirus capsid even after transduction by that covalent linkage, and thus it might not properly traffic to the nucleus for gene expression. The thing that AAV needs to do is adenovirus just goes to the nuclear pore and squirts its DNA through. AAV needs to sort of go around and go through the nuclear pore and then traffic through the nucleolus actually in order to uncode. And so I'm guessing that the efficiency could potentially be improved by using a endosomal protease cleavable linker, such as a GFLG cathepsin cleavable peptide, in the middle of the bycatcher. And that's a fairly easy hypothesis to test, and I'm planning on working on that soon. Before I move on, I just do want to point out that although there is somewhat limited co-expression here for my very first experiment, you definitely do see some examples, particularly in this field of view 2. There's several cells that very nicely line up together with co-expression. There's also a couple in field of view 7, field of view 5, and field of view 1. And these were randomly selected, so I actually just went around looking at green first, and then I just took whatever red was in whatever random field of view I picked from the green. So it's not like I'm biasing these necessarily. So yeah, as I mentioned, this part of it is still in very early stages, but I'm excited because I think there are definitely a lot of options to explore, and I'm currently actively working with a AAV expert who works at UAB to get advice on how to proceed and what steps might be beneficial to take. So from there, I'll move on to explaining what I plan to do. So I'm planning on demonstrating delivery of 2 AAV cargoes. So right now I just have the adenovirus and 1 AAV cargo, but the production of AAV encoding a blue fluorescent protein, MTAG BFP2, is underway at the viral vector core here at WashU. I provided them with the plasmids, and they're going to be making the AAVs for us. they're also making more TD Tomato AAVs since I'm running a little low on those. And essentially what I plan to do is make a one-to-one mixture of the blue AAV and the red AAV, and then use those to make add AAVs that encode essentially both proteins in addition to the GFP in the adenovirus. And then I'll test cells for co-expression of all three fluorescent proteins, and hopefully I'll be able to, with some further optimization, show that this is feasible in permissive cell types, and that'll be sort of a final figure for the first paper that I'm going to be working on as part of my thesis, the first data paper anyway. And so this is my plan for the near future. In the a little bit more distant future, I mentioned CRISPR at the beginning of this presentation. I have some more detailed information that just due to time I'm not going to cover, but essentially what I plan to do is make AdAAVs that encode Cas9 or another Cas protein, as well as two distinct guide RNAs, and then have AAVs that encode donor templates, probably for red and green, or sorry, red and blue fluorescent proteins, but potentially also for maybe some more disease relevant genes that could be tested in an in vitro model. And then what I want to do is show CRISPR mediated insertion of those two genes at target sites within the genome of cells. And that'll be the sort of the second big point as part of my thesis. I also have some other projects that I mentioned that might serve as additional papers too, so I'm excited to pursue those as well. So with that, I will wrap up my presentation with acknowledgements. I'd like to thank the Curiel Laboratory, all these folks, nice folks pictured here, our collaborators at Baylor and Rice, as well as the folks working at the core facilities at WashU, in particular Wandi Beatty, who works in the EM imaging core facility, has been just absolutely wonderful, very lovely person, and she has done a lot of great work for in terms of helping me capture the electron microscopy images, which has served as a crucial part of my project. And so I'd like to thank her in particular. And with that, I will conclude my presentation and open the floor to questions. Thank you very much, everyone.
Great talk. Thank you. Thank you. Before I looking at any question, Nathan, do you have any comment or question to him?
Yeah, I for sure have a question. I definitely want to have like the other attendees get the majority of the time, but maybe one question to Logan. So some of the choices that you're making now in terms of how you're implementing this could have downstream consequences. Like if this is ever going to go into industry or somebody's actually going to have to fabricate these things.
Mm-hmm.
So for example, it seems like you're using capping agents. Like you're labeling everything and then you're capping to decrease like the numbers. Would there be other approaches that you could take that might be better for downstream processing, purification types of things if industry were ever to pursue this?
Oh, absolutely. So yeah, so part of the reason that I'm employing the choices I'm employing now is because I'm really trying to uh, move relatively efficiently towards a good initial proof of concept, where I can validate this in cells, work through the challenges of validating it in cells, and so on and so forth. But there are a lot of options for both the capping and for the purification that I think would be potentially maybe a little more complicated to do right now, but as mentioned, I'm sort of looking into moving in that direction. But might be very beneficial down the road. So, for example, the biotin streptactin interaction is the streptactin column is actually originally intended to be used with a strep tag. So it does weakly bind biotin relative to streptavidin, but it still binds biotin more strongly than I would like. And so this may be part of the reason that there's somewhat limited yields. So finding affinity reagents which are more optimally suited to this vector may be highly beneficial. And really depending on because of the versatility of having all these SPY tags, you could have SpyCatcher fused to a wide variety of different affinity reagents, or you could even target the virus capsid itself, the adenovirus capsid, with some kind of affinity reagent. So there's really a lot of possibilities for improving it there. In terms of sucrose gradient purification, that is potentially a more efficient way to do it in an academic context. But in the industrial context, typically ultracentrifugation does not scale very well. And so it would be likely that a affinity column would still be necessary. However, I do think that there could be vastly improved affinity columns given additional resources and time and effort applied to that part of it. As to the capping, yeah, so the SpyTag MVP is a commercially available reagent that is convenient for now, but I do you can fuse SpyTag to a variety of other proteins as well. So you could even fuse it to things that might act as an additional shielding reagent. You could fuse it to something that would increase the affinity to certain cell types, so maybe a nanobody or something like that, and thereby you could have even additional sort of functionalization that would be very beneficial for various applications of this vector.
Right. Thank you.
Any other question, Nathan? Okay.
No, I'm good. Thank you very much.
Thank you. So in that case, I want to ask question because I waiting for other people to ask question. So thank you for your very detailed explanation of all the procedure and then your the project. And I want to kind of think about out of this project, what would be the biggest challenge in the future to achieve your goal? I understand, some detailed method you just described very well, but I want to see where the field is going, and then to go there, what is the most challenging, problems, and if we solve that problem, everything just kind of quickly just go there.
Sure. So I think that I'd like to structure this in terms of stages of advancement. So first, I'll talk about sort of the idea of preclinical studies, and the essentially I'll say that the first big hurdle beyond what I've described in this presentation will probably be to do in vivo tests and to just make sure that this works well in the context of in vivo, because that's currently kind of an unknown. Based on a lot of the literature, the information that we know, the design that I put into this, sure, it should work well in vivo, but we don't really know. And so I think that doing, at the very least, some small scale in vivo tests to start, just to see the biodistribution and so on and so forth, toxicity profile, etc., would be very beneficial. And I think that that will be sort of the first major hurdle. In the longer term, what I'm envisioning is that so the challenge for the longer term might come from the variability in the construction of this vector being an issue for clinical applicability, because in the context of clinical application of viral vectors, even things like just AAVs on their own, it can be very costly and very difficult to deal with the inherent variability of the complex macromolecular assembly that is an AAV. And in the context of clinical actual application of therapeutics, you need to have very, very nicely consistent batch-to-batch effects, and you don't want to have a lot of like randomness in the equation, because then what you give to one patient might lead to a totally different outcome that compared to what you give to another patient. And there's a lot of regulations on that. And due to the inherent construction of AAVs, I do think that there may need to be some kind of framework developed for ensuring batch-to-batch consistency that might be kind of challenging. I do think it's doable, but I think it'll be a challenging hurdle to overcome in the future. So batch-to-batch consistency for clinical application.
Okay.
I think there's a question in the chat. Oh yeah, go
Yeah,
ahead.
that makes sense. And I'll ask question from Maya. Maya asked, my understanding is that viral capsid don't always have high assembly fidelity to begin with. And have you compared your assembly with the native?
So if you mean by assembly fidelity, I'm not sure exactly what you mean by that, but I think that if you mean that essentially that they are essentially that they don't assemble very well, that's not necessarily the case. I mean, capsid modified viruses are really the problem because the viruses in their native environment, which I have compared to via transmission electron microscopy, um, viruses in their native environment do assemble very well. They've evolved to do so. However, capsid modified viruses can sometimes assemble less well. However, that usually leads, the big issue there is that you can get lower titers. And overall, from what I've seen, is that the AAVs and the adenovirus that have been capsid modified with SpyTag both have good high titers to begin with. And so that's not too much of an issue. I'm not sure if does that answer the question, or is there any follow-up to that?
I guess we can. Oh yeah, she said that's yes.
Okay,
Thank you.
great.
Okay, so I wanted to close because I see my clock almost o'clock. So thank you all for joining and staying today. We'll meet again on April 13th, Thursday, the same time, the same Zoom link, while I'm still in Europe. We will have Professor Pete Hesser from University of Michigan and Professor Ratul Kodukari, a new professor at Iowa State University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I will stop recording. Thank you.